Well, welcome to our BioStime Institute for Nutrition and Care Symposium this morning. Thank you for joining us. We have an exciting program today on Osteopontin with Professor Esben Sorensen from Aarhus University in Denmark and Professor Cai Wei from Shanghai University in China. Today we focus our symposium on Osteopontin, a very exciting protein from human breast milk and also from dairy cow's milk. And we have the inventor, the discoverer of Osteopontin here with us, Professor Esben Sorensen, and Professor Cai Wei, who devoted a big part of his research in China on the effects and the clinical research, pediatric research on Osteopontin.
First, I'd like to thank the organizers and BINC for this opportunity to talk here about my favorite protein. To start from the beginning, what is so special about milk? And I would say, so to say, everything is special about milk. In all these years I have worked with milk, this white liquid continues to excite me. We continue to find a lot of new stuff in there, and a lot of things that we didn't expect to find there. And what's the reason for that? The reason is that milk is absolutely the only food that's made to be eaten. Everything else is something that we have either cultivated or grown or done something with, and then we have learned to eat it. But milk is evolutionary evolved to be eaten. So evolution have had all chances through thousands of years, millions of years, to put in all kinds of good stuff for us as mammals to thrive and grow. So an enormous source of natural food stuff that gives us a lot of good stuff. For instance, milk has been shown to show a lot of specific oligosaccharides, which you all know are very important for the development of the microbiota in the infant. And then afterwards, that microbiota is very important for, so to say, everything. Also, a number of fatty acids, sphingomyelins, and other lipids are present in milk, which are very important, for instance, in neural development, brain development, cognitive development. And then there are several milk proteins that are not only there as sources of amino acids or energy. They provide something extra, something that was not expected to be there, so to say, but evolution, by having to deliver a lot of amino acids for the growing neonate, it could as well put in other stuff there that could be released during digestion and then first have some kind of bioactive effects, and then afterwards it could be fully digested and used as amino acids and building blocks in the neonate. Also, many milk proteins transport minerals and vitamins. Here I have collected a number of both well-known and also probably not or less well-known milk proteins. At the left, in the red circle, you see all the major proteins of milk. These are the ones that are expected mostly to be there for nutritional purposes. That is classical nutritional purposes, that delivers the amino acids and the energy for the neonate. If you go to the right, in the green circle, there's a lot of peptide proteins that are present in lesser amounts, and these all have more, in my terms, more interesting properties. These are the ones that deliver biological signals or have very specific functions. You can see transcobalamin that transports vitamin B12. There's lactoferrin that you probably all know that had antibacterial effects and also transports iron. There's other proteins in there. EPV20 or Niemann-Pick-C protein is a new protein we're working with that transports cholesterol in milk. And then, of course, we have osteopontin. And osteopontin is really something. I have to say that because I've been working with it for 30 years, so there must be something in it, at least I hope. It was first discovered in bone in 1985, and it was named osteopontin in 1986 by a Swedish group that isolated it from bone material. And the reason why it's called osteopontin is from the osteo Greek, meaning bone, pontin from Latin, meaning bridge. So osteopontin is the component that makes us stick to our skeleton, so to say. It is the link between the tissue and the bone. But that's only one function of osteopontin. That was the first function that was discovered of osteopontin. That's why it's got its name, osteopontin. Now we have learned that osteopontin is expressed, so to say, everywhere in the body. It's present in all body fluids. It's present in all tissues, in the brains, in the kidneys, in the liver, in plasma, in urine. It has very important functions everywhere, and these are different functions. There are now 30,000 scientific papers. If you make a search on PubMed of osteopontin, you'll get 30,000 hits, and I can say that I will not go through all of them today. I will focus mostly on the ones that have to do with milk. There are a couple of hundreds on milk and osteopontin. It has different names. It was actually discovered in 1979, but at that point they didn't know it was osteopontin. Then it was discovered as a component that was upregulated when T cells were activated. So it was called early T cell activated gene 1. Then it's called uropontin for the form that's present in urine. There it's actually the major inhibitor of the formation of kidney and bladder stones. And then there's secreted phosphoprotein, also a relic from the time where it was only present in bone. And then there's lactopontin, which is the name for the form of osteopontin in milk. So I will use either the name lactopontin or I will say milk osteopontin. Most interesting is that it's present everywhere. It's involved in, so to say, everything. I usually call it a transformation protein because every time something interesting happens in the body, osteopontin is expressed. That can be both benign growth, it can also be pathological conditions. Osteopontin is either there as part of the disease or part of the cure against the disease because the immune system expresses high amounts of osteopontin. The most interesting thing is that there are 500 to 1,000 times more osteopontin in milk than in any other tissue or biological fluid. So it must be there for a reason. Nature do not make jokes. There's no reason to express such a highly bioactive protein in milk unless there's a specific function for it. Differences between the osteopontin and the milk osteopontin. There's a lot of those. It was identified firstly in 1989 in human milk, and then, as Hanno Kaponen said, I discovered in milk, in bovine milk in 1993, actually as part of my undergraduate work. There are many osteopontin isoforms, but there are only one gene for osteopontin. Osteopontin is encoded on chromosome 4 in the human genome, and there's only one gene. So no matter where you talk about osteopontin from kidneys or from bone or from milk, it's all from the same gene product. Though there are hundreds, if not 1,000 different isoforms of osteopontin. Same gene, thousands of protein. And the reason for that is that it is very, very heavily post-translationally modified. After transcription and translation, the protein is a lot of phosphate groups, a lot of glycosylations are attached to the protein. It's also prone to digestion by a lot of physiological proteases that can cleave one or more places in the protein, and then actually by doing that, it exposes other sequences to the protein that then gives it another bioactivity. And then I should say that the osteopontin now is produced from cow's milk, has been so for a number of years, and it's now marketed and used in infant formulas, and it was approved by EFSA March last year. You can see here this model I have down on this slide. I have tried to, is the cursor up here? Yes, it is. You see all the phosphorylations. They are not randomly distributed over the sequence of the protein. They are actually appearing in clusters of 3 to 4 phosphorylations spanned by regions of non-phosphorylated amino acids. And that's probably due to the action of interaction with calcium and hydroxyapatite and other minerals so that they can be bound by these clusters of phosphates. If you look in the center of the protein, you can see there are 3 to 5 O-glycosylations, and these are all placed very close to these sequences down here, the RGD and the Zwickler sequences. These sequences are sequences that can bind integrin receptors on cells. And these are, so to say, the powerhouse of osteopontin. These are the sequences that make osteopontin interact with cells, and actually that way might be eliciting signaling cascades or just bringing cells closer to the protein. Human and bovine osteopontin are structurally quite similar. Of course, they are from 2 different species, but all the structural important and biological important elements are conserved between the 2 species. You can see I have made an alignment here, the human on top of the bovine osteopontin, and the phosphorylations are marked in red and the glycosylations in yellow, and the integrin binding sequences are green and then we have a proteolytic site which is blue here. And you can see they are all present in both the bovine and the human form. And most interestingly, there's much more osteopontin in human milk than in bovine milk. Much more. You can see that here there's approximately 140 milligram a liter on average in human milk. I'll get a bit back to that. In bovine milk, it's less than 20 milligrams a liter, and in infant formula, standard infant formula, they're even less. due to the way they manufacture the formulas. So there's a deficit of osteopontin in infant formula if you want to mimic human milk. We have this protein that is highly bioactive in milk, but why? As I said, nature do not make jokes, they must be there for a reason. So what could be the reason? If we look at the structure, we can get some hints. For instance, I showed that we have these phosphorylations that are scattered or distributed in these groups. These could be there because they can then interact with calcium ions. It could also be iron, or it could be zinc or magnesium. Actually, we have shown that it can also transport these essential ions. But we also know that the caseins that are present in much higher concentrations, they also do that. So it might be a bit redundant to have such a special protein simply for transporting ions, but it can do that. Another more sophisticated function could be that osteopontin functions as an opsonin. That is a protein or a molecule that marks bacteria or other stuff that has to be removed by the immune system. We showed back 10 years ago, 15 years ago, that osteopontin could actually bind to bacterial cells, both in milk and outside milk, and then it could attract macrophages, and these macrophages then engulfed or phagocytized the bacteria. So osteopontin could be there as a marker to, so to say, clean up the milk and remove bacteria. A third theory and thesis, and as one that has been shown by many groups, is that osteopontin actually is a cytokine, a cell signaling molecule that interacts with integrin receptors, and by this interaction elicits some kind of signaling cascades. It's also been shown that osteopontin can actually be taken up into the bloodstream and go many places in the body. It's been shown that you can actually, after ingesting osteopontin, you can trace osteopontin to the brain. You can trace it everywhere in the body. So some part of osteopontin actually survives the digestive proteases, are taken up, transported to other places where it can have some function. Here we have shown that osteopontin can increase the expression of the interleukin 12 in intestinal cells. Interleukin 12 is an indicator of activation of the cellular innate immune response. There's been one major large clinical trial. I'll just briefly show this because I know Professor Cai Wei will go into more details about this study. It was made in Shanghai in 2013, and there were 4 groups of infants, 80 infants in each group, and they were fed either breastfed, they were giving a standard infant formula, or infant formula with 65 or 130 milligram osteopontin a liter, mimicking either 50 or 100% of what we found in average human milk. Then they tested, after several months, blood samples of these children, and they could see that levels of inflammation, inflammatory markers like TNF-alpha and interleukin 10 were significantly lower in the osteopontin 65 and 130 group than in the reference group after 4 months, which suggests that osteopontin actually downregulates inflammatory signals in the intestine. There were also more regulating T cells in the infants that received the infant formula with osteopontin than in the reference group. And most interesting, especially from a parent's point of view, there was a significant lower incidence of fever through the 6 months period in the experiment in infants that received infant formula with osteopontin than infants that received the standard infant formula. Actually, there was not a significant difference between the days of fever among the breastfed and the ones that received the infant formula with osteopontin. Again, an indication that osteopontin does something to the immune system that's good for the baby. It lowers the inflammation, and it also decreases the days of fever. This is a mouse study where a mice model was induced with dextran sulfate sodium, which induces colitis conditions in the mice. And then the mice were treated with either osteopontin from milk, recombinant osteopontin, or control group, or simply water. And here you can see that after 3 days after the induction of the colitis, diseases start to develop, but you can see a significant difference. The mice that received the milk osteopontin have significantly lower disease index than the one that received both the water and also the ones that received the recombinant osteopontin. And that's why it's so important that we use the milk osteopontin, because all these modifications, they protect the osteopontin molecule so it's not fully digested, so it can still have its effect once it reaches the intestine. So here's an example that it's not always that you can just do, for instance, precision fermentation and do a recombinant stuff and do this protein, because all the good stuff in proteins, I must have say that as a protein chemist, they are attached after the translation and transcription. They also measured pro-inflammatory markers, and here you can see actually that after the ones that received the osteopontin, their levels of pro-inflammatory markers are almost at the same level as the control, whereas the ones that received none of those had very high inflammation. Another study here on osteopontin and atopic dermatitis, a mice model. Mice model was treated with a dinitrochlorobenzene at a shaved part of the dorsal area of the mice and also on the ears. And then this treatment mimics skin conditions that you see in atopic dermatitis. It was treated over 27 days, and every second day the treatment was done again, so that it had continuous these atopic dermatitis-like conditions. And then it was given either osteopontin in the diet, or it was given 2-fucosyl lactose, a major oligosaccharide from human milk, or a combination of the two. And then they followed the development of the atopic dermatitis symptoms here. And if you could see here, I need my glasses to see what I'm showing you. You can see that the levels of TNF-alpha, interleukin 6, interleukin 1 beta, and TSLP, they are all decreasing significantly after the osteopontin and fucosyl lactose treatment. Also, the number of eosinophils and mast cells, which are indicators of this atopic dermatitis, they are also decreased significantly. And overall, in a scoring of these mice in both the thickness, the scratching behavior, the general fitness, and the well-being of these mice, you can see that they are all going down after the treatment with this fucosyl lactose or osteopontin, or the combination of those. But to sum up with my conclusion, a very, very provocative conclusion, could it be that osteopontin is a or the missing link between the infant formula and the breast milk? We know that in breastfed children, they have fewer days with fever, they have less infections than the ones that receive the infant formula. So we want to mimic that. We know that osteopontin now affects the immune system, and that actually gene expression and cellular studies show that we closes the gap with between the human milk and the infant formula by adding the osteopontin, because it's present in high concentrations in mother's milk, as shown here, very high concentrations in mother's milk. And we have shown that these are very similar, so that bovine osteopontin can substitute for human osteopontin probably better than recombinant human osteopontin can. At least that was I claim, until I see something proved the other way around, because the post-translation modifications are very, very difficult to do in precision fermentation. And it has been shown that osteopontin reduces days of fever and lowers the inflammatory responses. So it could be a missing link, meaning that we could close the gap between the standard infant formula and the breast milk by adding osteopontin. And with this, I would like again to thank the organizers of this symposium. And if you want to know more about osteopontin, I'd like to draw your attention to either our review from last year, Milk Osteopontin and Human Milk, or the EFSA report that was written on osteopontin last year. And then this week, I've been part of a new panel that publishes a new report on the use of osteopontin in infant formula. It should be published in Frontiers of Nutrition this week or next week. And with this, I would like to thank you for your attendance.
Thank you, Professor Sorensen, for introduction. So first of all, ladies and gentlemen, first of all, I would like Bystone invite me to come here to communication with participants and other professional staff. So today, my presentation will be optimizing osteopontin in infant and children health. What's new? Fortunately, Professor Sorensen, she did a very nice presentation previously, so I my presentation will be more focused on the clinical practice. Th1, Th2 balance, when the body is in normal status, the number of the Th1 and the Th2 cell is dynamic balance, remaining normal cellular and humoral immune functions. When the body functions abnormally, the balance is skewed toward one of the two sides, which is called Th1 and Th2 drift, which will be leading emergence and the development of some disease. So, immune balance in newborn is skewed toward Th2. So we should be enhance Th1. When immunity is skewed toward Th2, some common allergic disease like atopic dermatitis, allergic asthma are likely to occur. Rapidly development of the Th1 immune response after the birth is needed to restore the Th1 and the Th2 balance, especially in the first of year life. So, optimal protein can induce the expression of the Th1 cytokine interleukin 2 and interleukin 12 and inhibit the production of the Th2 cytokine interleukin 10. Therefore, that regulated the Th1 and the Th2 immune balance. One from my group is the material and infant cohort study, correlation between osteoprotein content and the maternal composition of the breast milk in China. We divided two parts study. One longitudinal study, and the other is a cross-section study. Longitudinal study, we include 106 mothers. They are breastfeeding within 14 days or early postpartum, live in Shanghai during the pregnancy at least 6 months. We take the time of the breastfeeding collection and the follow-up. The timing are 1 to 14 days. The second point, 2 to 4 months. And the third one is 5 to 7 months. This is for the longitudinal study. The cross-section study, breastfeeding mother in the lactation age of the maternal breast, which including body composition analysis, blood sample collection for leptin and insulin, breast milk collection. Osteoprotein level are related to the maternal body composition, especially content of the skeletal muscle mass. Collection analysis showed that optimum protein was positively correlation with body weight, bone marrow content, skeletal muscle mass, body fat, BMI, and the visceral fat area. After the controlling the lactation time, all positive correlation between the above indicator was found was significantly different. In the mother, if the mother is a good health condition at the time of the breastfeeding, based on the weight factors and the human composition, therefore, changes of milk referred to the optimal in the study will significantly benefit for the kids. Thank you.